So today I'll talk about the sleeping beauty cancer cells, also called tumor dormancy. So this is when cancer cells remain alive but don't grow. So the patient appears disease free, but months, years, or even decades later sometimes, the cells wake up and cause recurrence. So some examples are ER positive breast cancer can recur 20 to 30 years later, melanoma can recur 10 to 20 years later, renal cell carcinoma may recur decades after surgery, and prostate cancer can remain dormant for years. So, where are these dormant cells hiding? They are hiding in the bone marrow, the lung, the liver, the brain, the lymph nodes, perivascular niches. So, there are three main types of dormancy. One is cellular dormancy. So, here single cells stop dividing, the cell will enter a resting state, and cells are alive but inactive. So these are sleeping cancer cells. The second type is tumor mass dormancy. So here the cells divide, cells also die, growth and death remain balanced. So the result is tumor size does not increase. And the third one is immune dormancy. So immune cells continuously suppress cancer cells. So T cells keep the tumor under control. If the immunity weakens, recurrence occurs. So how do cells survive so long? So some of the reasons are, one, they have a low metabolism. So they use less energy, less protein synthesis, less dividing. So the cell enters a power saving mode. Two, stress resistance. So dormant cells can tolerate hypoxia, low oxygen conditions, lack of nutrients, chemotherapy, oxidative stress, everything. 3. Stem cell-like properties. So many dormant cells would show self-renewal, therapy-resistant, stem cell-like behavior basically. And 4. They form protective niches. So bone marrow and other tissues can give them growth factors, survival signals, and immune protection. So what are important molecular pathways that help these sleeping beauty cells? So one, high P38, which promotes dormancy, and low ERK signaling. ERK reduces proliferation, cell division. 2. TGF beta signaling. This can help induce cell cycle arrest, so maintains a quiet state. 3. NR2F1. So this is a dormancy regulator. It's associated with long-term quiescence. And 4. Integrins and extracellular matrix signaling. So interactions with extracellular matrix help maintain dormancy. But then why doesn't chemotherapy kill these cancer cells? So most chemotherapy targets dividing cells. I've spoken about this before. Dormant cells are not dividing. So therefore they can escape chemotherapy, and chemotherapy does not affect them, and therefore recurrence can occur. But then what wakes these dormant cells up? So the possible triggers are inflammation, aging, immune decline, changes in blood vessels, tissue injury, fibrosis, changes in the extracellular matrix. The exact trigger is often unknown, but these are the possibilities. Now, why is studying dormancy so important? Because this phenomena explains late recurrence of some patients, why some patients had recurrence of their cancer after 20 years, even though they thought that they were disease free. This also explains minimal residual disease, some of the disease that might be remaining. And this is the major cause of treatment failure. And it's an important target for future therapies. Therefore, it's very important to study.